the Congress for inviting me for this speaking invitation, and Kate Broderick did give the disclosure previously that I was at the University of Pennsylvania when this work was completed, but it was done in collaboration and funded by Inovio, and I'm now currently an Inovio employee for several months. So as I'm sure everyone is well aware, malaria is still a significant global health concern, and actually over half of the world's population lives in malaria endemic areas and are at risk of malaria. There's actually been a recent decrease in the amount of deaths and incidence of cases because of greater infiltration of preventative measures into at-risk populations. But there was still over 500,000 deaths last year, and most of those are in young children. So in that vein, an effective malaria vaccine could be a key tool in reducing the global health burden of malaria. So why don't we have a malaria vaccine yet? Is a malaria vaccine feasible? And actually, one would think that it is because in endemic areas there is an acquisition of naturally acquired immunity. And we have shown in studies in animal models and in humans that using sporozoite-based vaccinations can confer protection. But there are multiple challenges in malaria vaccine development, including that it's a protozoan with a huge genome. Unlike a virus, it has 14 chromosomes. It has a multi-stage life cycle, which I'll touch on in a second. And there's also high levels of antigenic and allelic variation in target antigens. The other issue is that we really don't have a defined correlate of protection. It could be antibodies, it could be T cells, or it's probably both. And there's also the lack of a good animal challenge model because the strains of malaria that infect mice don't infect non-human primates for the most part. And those strains also, for the most part, do not infect humans. So there really isn't a good clinical model for us to test out novel vaccine candidates. So the malaria vaccine I'm going to discuss today touches basically on two stages of the malaria vaccine life cycle, the sporozoite stage and the hepatocyte stage. So anti-sporozoite vaccines are, for the most part, they are trying to generate very high titer antibodies to prevent the sporozoites from migrating into the liver. This progress only takes about the maximum of 2 hours, so you have a very short window and need very high affinity antibodies. The other approach is targeting the hepatocytes once they are infected. And again, it's also like looking for a needle in a haystack. You need good CD8 responses for this. So these are the two different life cycle parts that I'll be discussing. And many vaccines, including the one that we are working on developing, target multi-stages. So we're trying to induce high affinity antibodies as well as strong CD8s. So two of the most farthest along leading vaccine candidates to date are one that's based on sporozoites, and the other is RTSS, which I'll discuss shortly. So in the 1960s and 1970s, in animal models, mice, and also in humans, it was shown that you could vaccinate with radiation-attenuated sporozoites and confer protection. A more recent study showed that using live sporozoites in patients undergoing chloroquine treatment could also confer protection from subsequent infection. But there's one big issue with sporozoite-based vaccines, the key being wide administration in the field. So another group called Scenaria, which is based in Maryland, developed a large-scale-ish production method and did some studies looking at IM and ID administration of this large-scale production of sporozoites and saw minimal efficacy. But then, in a subsequent study, they showed that you could give 4 to 5 doses IV and actually confer protection in humans. So this was a great proof of concept and quite a large step forward for the malaria field. But again, how are we going to administrate this to young children in the middle of Africa and give them 5 IV injections? So it was great for proof of concept, but there is still a need for further malaria vaccine development, such as RTSS, which is in phase 3 and has showed 50% efficacy in 5 to 17-month-olds, as well as 30% efficacy in 6 to 12 weeks old. It's based on the circumsporozoite protein, which is a coat protein of the sporozoite. And what we have learned from the RTSS vaccine development is that antibodies directed at the circumsporozoite protein, or CSP, are associated with protection, but they may not be sufficient. And that CD4 T cell production of interferon gamma and IL-2 may also be associated with protection, but not sufficient. But RTSS only induces CD4 T cell responses, and a lot of research has shown that CD8 positive T cell responses are probably very important for clearing malaria infection. So this was the first results that were released from the RTSS vaccine phase 3 trial. And this top line here is the control vaccine, which is a rabies vaccine. And here is the RTSS vaccine looking at efficacy over time. So you can see again that there really is a need for an approved vaccine targeting malaria. And this has been covered several times in the morning session, so I am not going to belabor DNA vaccines and why they remain a promising platform. But I think that this next figure on this slide really shows that the issue with immune potency. This study was done in the 1990s by Merck. This is an interferon gamma alispot, looking at GAG-specific responses in non-human primates. You can see here that DNA was on the low end of the scale, and our goal then became to try and reach T-cell responses that are equivalent of those induced by the Ad5 platform. So as has been discussed, electroporation was one of those techniques to increase immune potency. There's also been things like molecular adjuvants and improving the antigens themselves and optimization, leader sequences, and things like that. This is an example of a study that was done in mice showing the difference that electroporation can make in both T-cell responses by interferon gamma alispot and also antibody responses. So, and Kate did mention this earlier, and these results were recently released, and we were all very excited about Inovio and in the DNA vaccine field, showing that the results of the proof of concept HPV phase 2 trial. It was a placebo-controlled randomized double-blind trial with 150 approximate subjects. The primary endpoint was regression of SIN2-3 to SIN1 or to no disease. And our secondary endpoint was regression plus clearance of the HPV 16 or 18 genotype that was shown at the initial stage of the study. These women received 4 different immunizations at week 0, 4, and 12. And we did reach both primary and secondary endpoints, the primary endpoint being regression. So there's a statistical difference in regression with VGX 3100, which again was targeting HPV 16 and 18 to those receiving the placebo control. And very impressively was that we did see, compared to the placebo, in the patients that received VGX 3100, there was a very large decrease in the amount of HPV 16 or 18 that was present at the end of the study. So the idea for the malaria vaccine was to take this DNA vaccine approach and select several different sporozoite and liver stage antigens. First, look at immunogenicity in the mouse model. And then I'll discuss briefly protection studies using a chimeric sporozoite challenge model, which allowed us to actually look at challenges in a mouse model of malaria with the CSP antigen, and then to look at immunogenicity in non-human primates. So this project was started in collaboration with MVI. With them, we selected 4 different antigens. 3 of these had previously been in the clinic. Celtos, which is still in an ongoing clinical trial, is the only one that does not have clinical results, but it's a highly conserved protein. They really felt it was a good target, so we went with that. CSP, as I mentioned, is the target of RTSS. TRAP has been used in the clinic with some success by Adrian Hill's group at Oxford. And LSA1, which I'll discuss a little bit further, is a liver stage antigen. The clinical trial using LSA1 used a recombinant protein-based approach. So LSA1 expression increases as the liver stage progresses, and therefore inducing CD8 responses to LSA1 may provide more efficacy than the antibody responses one would expect with a protein-based platform. So we decided to look at LSA1 despite the previous negative results in the clinic. So of these 4 antigens, CSP and TRAP were the least conserved. So we generated consensus antigens, which basically what you do is you take the all available full-length sequences in PubMed and use the most recurring amino acid at each different position in an effort to increase the breadth of the immune response and hit as many epitopes and as many strains as possible. So we did that for CSP and TRAP, but LSA1 and Celtos are highly conserved, so we just went ahead and used standard sequences for that. So the idea between selecting these 4 different antigens, for CSP, TRAP, and Celtos, they're sporozoite coat proteins. So we wanted to induce high antibody titers to decrease the amount of sporozoites that got to the liver and then T cell responses against all of the 4 antigens. So the first thing we did was look at immunogenicity in mice. In biopsy mice, they received 3 immunizations 3 weeks apart. And we looked at T cell responses and humoral responses by interferon gamma ELISpot flow cytometry, and ELISA at week 7, so 1 week after the last vaccination, and week 16, which is 10 weeks after the last vaccination. So the mice either received 30 micrograms of the individual antigens or all 4 in combination, which returned MAV4. And again, this vaccine was delivered using the Inovio Selectra constant current device. The Selectra is an IM device in the mouse model, but it would be ID in larger animal models. This is looking at antibody titers in the mouse model. These are just representative titer curves by ELISA. And here we're looking at antibodies to CSP, LSA1, TRAP, and Celtis. One thing that's been noticed in the malaria vaccine development field, as well as other vaccines, is that there's some immune interference when you co-deliver different constructs together. And this is particularly important for the circumsporozoite protein. So in this case, we did not see a decrease in antibody titers between CSP delivered by itself and MAV4. The other thing we wanted to look at was if the antibodies that were generated, if we could overexpress them in the liver of mice and then use sera from mice that were immunized with individual constructs and detect these antigens in vivo. So basically what we did was give the mice a tail vein injection using the same constructs that they were immunized with, and use sera from immunized mice to stain different liver sections, and you could see we could detect CSP, LSA1, TRAP, and Celtis antigens in the liver that way. Looking at T cell responses, this is by interferon gamma ELISpot. Again, we looked at all the antigens by themselves. And then as MAV4, the combination 1 week after the last immunization and 10 weeks after the last immunization. And I should point out that clearance of liver stage parasites is thought to be primarily dependent on, obviously, T cell responses. So we were very happy to see here in the CD8 T cell compartment that we really didn't have a decrease between 1 week and 10 weeks after the last immunization. And importantly, that most of the responses were interferon gamma positive or TNF alpha and interferon gamma positive. So the decrease we see in the ELISpot here is most likely due to the decrease we see in CD4 responses here. So this is great. We could detect T cell responses in the spleen, but what's going on in the liver? So what we did was we immunized the mice with the MAV4 cocktail and isolated splenocytes out of the liver and looked at CD8 interferon gamma responses by flow cytometry for 2 of the 4 antigens. And we could see responses to both CSP and LSA1 in hepatic lymphocytes, indicating that when we immunize in the periphery, we can still induce interferon gamma responses in the liver. So switching gears a little bit, in collaboration with Fidel Zavala at Johns Hopkins, he has developed chimeric parasites. So what these are Plasmodium burgii sporozoites, which will infect mice at the same rate as regular wild-type burgii sporozoites, but they contain the Plasmodium falciparum circumsporozoite antigen. So we could immunize using a falciparum CSP antigen and challenge these mice and look at parasitemia levels in the liver. So he also identified this novel CD8 epitope in the N-terminus, and then this is a known CD8 epitope in the C-terminus of Plasmodium falciparum. So we used these 2 models with just the C-terminus and the N-terminus and the C-terminus. We immunized the mice using our standard immunization scheme. And then 2 weeks after the last immunization, we challenged them with these chimeric sporozoites. So 48 hours post-challenge, we also looked at T cell responses in the spleen. We couldn't use the livers because we needed those for the challenge. And you see we have very strong CD8 interferon gamma production for the most part, both to the entire antigen and just to this novel N-terminus epitope. And again, most of the responses were either double positive gamma and TNF alpha or interferon gamma, which are responses that have been associated with parasite clearance. So more importantly, what we did see was a decrease in liver stage parasitemia compared to naive. So liver stage parasitemia is measured by qPCR. And this is in immunized mice. in naive mice, and there was actually a statistical significant correlation between the level of CD8 T cell responses and the levels of parasitemia. So the higher the CD8 response, the lower the levels of parasitemia in the liver. We also saw a similar thing in the C-terminal model. So again, in immunized mice, there was less parasites than naive mice, and an inverse correlation between the magnitude of the CD8 responses and the number of parasites in the liver. So to move forward with this hypothesis that the parasite clearance was probably due to CD8s, we depleted the mice of CD8s. So we had a group that were immunized, a group that were immunized and then CD8 depleted, and naive. You can see that in the group that had their CD8 positive T cells depleted, there was an abrogation of any type of protection from immunization, indicating that at least CD8s play a very important role in parasite clearance in response at least to this epitope. So almost done. But importantly, how does this translate into the non-human primate model? We used the MAV4 construct and immunized rhesus macaques 4 times. At first, it was 6 weeks apart, and then we waited and gave them a 4th immunization at week 25. So they received 1 milligram of each of the constructs, delivered IM with EP. And we saw IFA binding titers. So these are titers where the sporozoites where the antibodies are used to show that you can specifically bind to the sporozoite, so it's a functional antibody. And then we saw binding titers to CSP, LSA1 trap, and celtos. That, for the most part, did boost with each immunization. And then looking at T cell responses in the non-human primates for interferon gamma elipsot, again, we saw a good boost with each immunization. And then in the CD4 compartment, in the CD8 compartment, between the 3rd and the 4th immunization, we didn't really see a boost in the total CD4 response, but we did see a shift in the population. And then in the CD8 compartment, something very similar, we saw like a shift in the population between the 3rd and the 4th. But importantly, this is one of the only studies that has shown a combination vaccine delivered this way where you can still induce CD8 T cell responses to the CSP antigen. And last but not least, that's great, we have CD8 T cell responses, but did they do anything? So we then looked at antigen-specific granzyme B and TBET as looking at potential effector in CTL function. And this is antigen-specific granzyme B, antigen-specific TBET, and we did see an increase up to the 4th immunization. And the same thing here, and these responses did not decrease for at least 12 weeks after the last immunization. So it looks like our CD8s may actually do something. There's further studies in progress to see if these can actually kill target cells. So I'm short on time, so I will eliminate the summary, and I would just like to thank David Weiner's lab at the University of Pennsylvania, where this work was conducted. Rebecca Lee and Amrita both worked under me for a couple of years and really helped out with this work. Fidel Zavala's group at Johns Hopkins, and of course Synovio. We were funded in part by PathMVI, and Walter Reed did some of the IFA binding assays for us. So with that, I'll take any questions.
Thank you, Bernadette.
Did you, in your VA vaccination schedule, did you add any genetic adjuvants like?
We did. I just, due to a time limitation, I did not show that data here. We actually looked at IL-28 as a molecular adjuvant because it's been shown to increase CD8 effector function and CTLs in some other studies. And we did have some interesting results using IL-28, and that work is actually, the publication's in progress at the moment.
Yes, so I have a question. There have been a number of studies where DNA immunization has been used for malaria with little success. I know that electroporation was not the method of delivery, but have you thought of improving or combining DNA immunization in a prime boost strategy with another, rather than just as a standalone DNA vaccine?
Yes, we have. We've thought about combining it. Tom Ritchie's group has been working on some viral vector-based, so we thought about maybe doing something like that. We've also looked at incorporating a protein boost. We did that in non-human primates as well. It did see that boost antibody responses more and the DNA alone at the end. So they're all things that have been considered, and we're looking into further. We're going to give.
So one last question right there.
So there was some interference with the CSP antigen and T-cell responses, but in comparison to some of the other studies, we still had a high enough magnitude where we weren't really worried about it. And so it basically was CSP, and everyone's been saying that, so we've been working on different